I wanted to get started and introduce our guest speaker for today, who is Dr. Mary Armanios, who is a director of the Telomere Center at Johns Hopkins. She's currently a professor of oncology, genetic medicine, pathology, and molecular biology and genetics at Hopkins. And since 2019, she's been the associate director for education and training at the Sidney Kimmel Comprehensive Cancer Center. And her research focus over the last 16 years has been understanding the role of telomeres and telomerase in disease. She has defined the spectrum and genetic basis of the telomere syndromes, and with her trainees dissected their underlying mechanism. So without further ado, I'll hand it over to Mary. And again, a big thank you for agreeing to be our guest expert.
Everybody, and thanks for joining. And it's a great pleasure to learn more about the ICARE efforts and how collaborative this group has been and what an impact it has been. And I had the pleasure of attending one of the meetings a few months ago, and I'm very, very glad to be back and to continue our conversation. So as you heard from Dr. Pal, I've been interested in telomere disorders in general. I'm trained as an oncologist, but I've been sort of following clinical clues to understand the spectrum and the patterns of this sort of special group of diseases. And I'll tell you why they're unique in many ways. So just by way of introduction, telomeres are repetitive ends at the ends of chromosomes. They have a tandem repeat T2AG3 sequence. They have specialized proteins. And for a long time, since the 1990s, they've been collected to cellular processes of aging, where telomere length shortens as cells divide. And eventually, at a critical threshold, cells can no longer divide because they senesce or die out. And this has been, flashy and a lot in the press in the 1990s and early 2000s. And by the time I ended up in my research postdoc, I was sort of skeptical if this meant anything for clinical care. And as you probably now know, these are important syndromes in multiple settings, including in cancer, but not only in cancer. So telomerase is the enzyme that synthesizes new telomere repeats back. It has these two essential components. One of them is TERT, the telomerase reverse transcriptase. And this enzyme has the ability to create new telomere repeats. It uses a special RNA that provides a template, and this reverse transcriptase can create new telomere repeats onto chromosome ends. It does that very cautiously. It does that in very limited windows of development, such that telomere length is determined during embryonic development and thereafter shortens. And therein lies the importance of genetically determined telomere length for the human population, but especially for individuals who are born with mutations in TERT, and in any other components that regulate telomere maintenance, as I'll tell you. So we're going to focus on TERT today, but there are multiple genes that phenocopy these disorders. So the first family that I studied was a family that ends up having a canonical, pattern of manifestations that I will just use for an example as we talk about our cases today. So the young man here came to a hematology clinic. He had bone marrow failure and needed transfusions to maintain his blood counts. He also went gray when he was 9, and he eventually was diagnosed with pulmonary fibrosis. But in his family, these features showed genetic anticipation, where blood counts became lower and lower with each generation. And more importantly was the observation that more people in this family had pulmonary disease than hematologic disease, and none of them had cancer. Unfortunately, even though I was a cancer fellow, I tried to explain to my colleagues that this was important, but Many of them didn't believe me because they had not developed cancer. So genetic anticipation, multiple phenotypes, predominantly in the lung, but also in the bone marrow, and really in the liver. And this family carried a mutation in the reverse transcriptase domain of telomerase and ended up being an important example of how telomerase mutation manifests in the human population. So I'm having a hard time switching to my next slide. Okay, thank you. So telomerase mutations have their most common manifestations not in the disease that is in our title today, myelodysplastic syndrome, but these patients are at risk for developing cancer. And many of you may have seen or may likely see these patients and be consulted on them because they don't fit easily into one disease category. There's usually not a pulmonary genetics clinic at most centers, and a lot of these patients are referred to cancer genetics just because they often have blood disorders concurrently. So pulmonary fibrosis and IPF in particular, the idiopathic form, is progressive with age, and it's highly familial, more familial and more autosomal dominant inherited than breast cancer and ovarian cancer even. And a large proportion of these patients end up carrying mutations in one of the telomere genes, the most common being our focus today, which is TERT. It explains 15% of familial pulmonary fibrosis and these other related genes that are components or lead to telomere maintenance, function and telomere maintenance in some way or another. And I'm going to give some examples today of TERT mutations, and we'll hear some about some as well from the cases presented, but I'm also going to mention the other ones just for the sake of completeness. Just to orient you again, the genes we are studying are genes that are involved in either the biogenesis or the development of this important enzyme, which lengthens the telomeres, or in genes that regulate proteins, that encode for proteins that regulate its recruitment or its effectiveness, basically, and enzyme activity. So you can basically explain a lot of MDS genetics and pulmonary fibrosis genetics by knowing a little bit about telomere biology. And important for our conversation that as we're going to talk about in the cases, and I'm going to finish in 5 seconds, is the association between pulmonary fibrosis, bone marrow abnormalities. This is a bone marrow biopsy. Here's a healthy bone marrow with all the purple cells being the cellular component of the bone marrow. And when the telomere is short, the bone marrow is prone to not being able to regenerate the bone marrow stem cell in particular, and it becomes replaced by fat. In the lung, though, instead of the lung being replaced by fat, becomes scarred. It shows this fibrotic phenotype that we talked about earlier called idiopathic pulmonary fibrosis, as well as an aging phenotype in the liver that is caused by liver cirrhosis. And the combination of these this triad defines the short telomere syndrome entity and is the most common of premature aging syndromes. It does not, these complications don't always occur. They appear in about 30% of cases, with the most common, in 10% of patients with adults with short telomere syndromes have the triad, but the most common entity is familial forms of pulmonary fibrosis. So this is the typical presentation with genetic anticipation in older adults, and then their children become successively more prone to developing hematologic disease. And this is unique because most genetic anticipation syndromes have been defined as an earlier and more severe onset of disease in the same organ. But here, the predilection of disease switches and becomes more prone to becoming in the blood. And this is canonical, but one can also see the pattern in this format where short telomere syndrome patients have variable manifestations. And this is what makes them challenging for us in genetics and in the clinic. These are the same disease with the same genetics. So it can be the same mutation in TERT, but in the parents and grandparents, it will present as lung disease. And in the children, it will present, and young adults, it will present in aplastic anemia or bone marrow failure. And then in infants and young children, it is more likely to be syndromic, a very rare manifestation. And in this group of patients, as we'll talk later, this is where the cancer phenotype is more likely to show up. And about 10% of patients with short telomere syndromes will develop MDS, usually co-occurring with pulmonary disease. So just to show you in another angle, TERT is the most common, with accounting for about 45% of all the Mendelian cases of short telomere syndromes. And we also have this telomere length measurement in the blood that allows us to query the functional significance of mutations. You see, this test has a very high negative predictive value. For their telomeres long and someone has a mutation, even if it's pathogenic, it will unlikely cause them any symptoms, but it may manifest later in their children and their grandchildren in the future. So this is my last slide, I believe, before I turn it over back to the ICARE team. This is what I want to leave you with and to think about with you as we think about the cases in the next few minutes. Familial pulmonary fibrosis with bone marrow failure in the same family is a diagnosis that can be made clinically, a diagnosis of short telomere syndromes, even without molecular testing or genetic testing. I think to a great degree, one can make this diagnosis clinically at the bedside with a good family history. And a mutation may be identified in up to 80% of these patients right now using the genes that we know of. We know that the rest of the 20% are genes yet to be discovered. Familial pulmonary fibrosis, this is a first-degree relative affected. It's about a third. So these are very high pre-test probability clinical scenarios. And then IPF all-comers is about 5%. MDS all-comers I will talk about later. It's a little bit approximately within the range of the sporadic IPF. And with that, I will stop here and turn it over back to Dr. Ross.
Great, thanks very much. Thanks, Mary. Thanks for letting me speak today. It's a very quick presentation. I had a discussion with Mark Tischkowitz, and I can see the link there in the previous talk. And he discussed that it might be useful to get some feedback. And hearing your talk, Mary, it seems like this is exactly the kind of place to get to discuss such a case. But just as a caveat that this is a case where it's a bit of an unusual one for us in that we weren't involved with this care of this patient at our hospital the entire time. So some of the care was performed at different hospitals, and therefore our information is slightly limited. We're trying to get more information, but it is limited. So we have a relatively simplistic view of, I guess, of the case. What we do have is ongoing dialogue with the family. So I'll crack on. This chap that we saw is a 49-year-old gentleman, and when we first saw him, it was because he'd been admitted to the ICU, I believe you call it in the States, the ICU with acute shortness of breath, but on the background of a 1-year history of worsening cough and increasingly developing a shortness of breath. Unfortunately, he then died soon after, so I was never able to actually talk with him because he was on the ventilator and then passed away. I was able to talk to his wife, and that's where most of the information comes from, although her knowledge of his family is relatively limited for personal reasons. So we've tried to cobble together what we can. But he's got no exposure to industrial chemicals. There's no environmental suggestion of a cause for pulmonary fibrosis. He was a smoker and had stopped smoking, not due to any symptoms of shortness of breath, for 14 years prior to his diagnosis. Something he'd always noted, and that his partner had always noticed, was that he had premature onset of hair graying, which was unusual in his family. But otherwise, no unusual features and no other significant medical history. This has come across, I don't know how big this is on your screens, but here's our proband here. If you can you see my, no, you can't see mine because I'm not showing them, you're showing. You can see that the arrow there marks out the proband as deceased, and his 5 children, 2 of which are twins. And his father passed away in his 50s with cirrhosis of the liver, which his partner believes was secondary to alcoholic liver disease. And very little is known about the mother, but she also died in her 40s or 50s, we believe, and we know very little about her family. And the father of the proband came from a large family, but again, our knowledge of that is really limited because there was little connection with the family. So the wife, unfortunately, didn't know great details about them. She is in contact with the 3 sisters of the proband, all 3 of whom are, again, heavy smokers, and all in their 40s. One of them was admitted to the ICU about 9 years ago, so at the end of her 30s. She required ventilation for 2 weeks. And again, our knowledge on this is a little bit limited, but we understand that there's a diagnosis in her of chronic obstructive pulmonary disease, again, onset in the 40s on the background of smoking. And we've tried to contact this sibling of the proband to try to understand a bit more about her diagnosis and try to understand if COPD is actually the correct diagnosis there, or if something else might be going on, or there might be a dual diagnosis. These children, the 5 children, they are 3 of them are over 18, and the eldest, unfortunately, is a heavy smoker already in his early 20s. None of them display any signs of premature aging. So we, at that time, so things have changed in the way that we test in the UK now. But at that time, we were able to do a sort of a focused panel of pulmonary fibrosis genes. We used the Gemini panel for that. That's all that testing has now been replaced, but that's what we did at the time. And we were fed back this variant of uncertain significance in TERT. So it's a novel variant. We couldn't find it in any databases. It's absent from both NEM and ClinVar. It's in exon 10. There's this polar to non-polar changing of the amino acids. So it's obviously a missense variant. Doesn't appear to be any specific binding domain that I can understand from looking at the protein structure. And in silico does seem to be predicted as being deleterious and probably damaging by polyphen. We tried to contact some colleagues in other centres to try to understand if anybody else might have one that hasn't been published in a database. And one of the centres we reached out to did have what seems to be a similar, if not the same variant. We're just double checking that. And also on the background of very early onset pulmonary fibrosis in a female proband. But that dialogue is just open, so that's a bit of an open chapter. So I had a chat with our chaps here in the lab. Unfortunately, they weren't able to pull up any of the old blood films, to analyze any of the old blood films. None of them were available. And we weren't able to do telomere analysis. I'm not sure how it is there. Here, it's not a routine test that we would offer. I approached a group in King's College London, but that no longer seems to be an option. So we're as it stands, unable to assess telomeres for this patient. As I said before, we've made an inquiry through the spouse for the affected family member of COPD, so I try to explore that diagnosis and try to uncover whether there might be something else happening with that family member. We haven't engaged with the children as yet, but we've, via the partner, via the widow, we've tried to obviously encourage smoking cessation. Good advice in any background, particularly with this one, and no genetic testing at present. So our real quick questions here on how to proceed with this case is whether anyone, well, we're pretty open actually about any feedback, because we find this case quite troubling. But whether specifically what we're trying to understand is whether there might be any options for telomere testing or any other appropriate surrogate test, and what people feel about how we might proceed with the family, because although it's difficult to access the records of the deceased parents, we have 3 siblings who are all alive, and there are the 5 children, 3 of who are of adult age now. So I'll stop there. Thank you very much.
Thank you so much. What a great workup. Would you mind if we went back to the pedigree? I guess I have control over the slides now, so maybe if it's okay, I'll go back to the pedigree here. So I think this pedigree, in terms of what we were just talking about, IPF in a young person, that increases the yields, and I would call this, a familial form of lung disease, even though the sister has emphysema, and I'll talk about that in a second. Father with cirrhosis attributed to alcohol in the setting of someone else who's young with IPF is classic for what we see in short telomere syndromes, Dr. Rosso. In the setting of the mutation you found, which is in the reverse transcriptase domain, even in the absence of telomere length testing, I'm going to say this family has likely diagnosis of a short telomere syndrome attributed to a TERT mutation. I think I would treat them as such. In terms of just a word about telomere length testing, I oversee the only lab in the US, as far as I know, for telomere length measurement, and we've analyzed over 7,000 samples so far. We help regularly folks from overseas because this test is difficult to obtain. There's, I think, a lab that used to be in Switzerland, but we're happy to help you if you just email me and we can process a sample for someone in this family if needed. The interesting thing to note and why this family so offers so much to learn is the classic history of COPD. We have described emphysema in smokers with telomerase mutations, and it is the second known Mendelian cause for severe emphysema in young people. And it interestingly happens to arise more often in females than in men. So men who smoke most likely get pulmonary fibrosis, but women who smoke are more likely to get emphysema associated with pulmonary fibrosis or emphysema alone. So this history is not atypical, and you can diagnostically try to classify her lung disease, but she has a telomere mediated lung disease, and we've tried to group these disorders because they're very hard to classify actually. And I think for us in genetics, it's just if a genetic cause attributes to the risk of all of these disorders, we might as well combine them. I'm going to guess that your patient, the proband who unfortunately died at such a young age, may have had blood count abnormalities that may not have been noted. I'm not sure. He didn't
No, I mean, so again, he was, when he was treated in the ICU, it was in our heart and lung hospital, which is separate from where we are. So I haven't got access to his most recent, but going back historically, his blood counts were relatively normal. Going back a couple
So
of years.
sometimes we can, that's great. You know, the most common subtle abnormalities sometimes tend to be macrocytosis, which is enlarged red blood cells, which sometimes is a sign of stress in the bone marrow. But I'll say that, you know, when IPF is diagnosed in TERT mutation carriers, or let's say all of the telomere gene mutation carriers under the age of 50, at least half of them have a blood count abnormality. And I will just tell you that, you know, we were one of the first groups to describe this premature graying. That's also, you know, a reassuring sign that we have the right diagnosis here, although it is non-specific. And if you were to test everybody you see for premature graying and lung disease, that's a lot of people and the yield is not high. But in the setting of this family history and this severe disease, and just Dr. Ross's clinical description of how acutely this patient died and how quickly and how unexplained is why this disease has been called idiopathic and why the telomere genetic connection has been so helpful in understanding its etiology. It really places it on the map as a genetic disorder that we can at least understand what causes it, even though we may not have as much to do about it as we do in cancer. So in terms of the children, you know, that's a matter of, I think, you know, what we do is counsel, and because of the genetic anticipation pattern I showed where children are more likely to develop cytopenias and hematologic disease in a setting like this, we often just check their blood counts to avoid making sure that they don't get treated with other, you know, drugs that may be harmful. So I think we counsel on don't get treated for any condition without talking to us if you don't get genetically tested. Let's check your blood counts. You know, sometimes these later generations are more likely to manifest some subtle phenotypes of dyskeratosis congenita, but most often not. And then the counseling about the smoking, which you mentioned. That's what we do in the most conservative setting. If they were tested and they have normal blood counts, I think we survey these kids very conservatively. And I'm going to talk about surveillance at a slide later. Maybe I can go to that. Should I keep going or? Yeah.
That would be great. And, you know, Mary, the other question I had for you as you were talking is, are we seeing, so at different ages, the risks are different for a different disorder, right?
Yes.
But are we talking about pleiotropy, like from a genetic standpoint? So the same gene can cause different manifestations and we don't know what's going to happen, and they're really not related manifestations? I don't know.
Yes, you can predict. I mean, this is where the telomere length is very prognostic, at least for those of us who stare at this and have seen so many patients. So let me address that. I think it's coming in the next slide or the one after, Dr. Paul. But I wanted to show you, you know, this mutation in the 700s, I think, is right smack in the middle. I can't see completely, but it's in the middle of the reverse transcriptase domain. So you see here, this is the TERT protein, and the middle part of it is where all the mutations lie. And these are classic loss of function mutations, usually missense mutations. And to Dr. Powell's question now, you know, let's look at the telogram more carefully. And this is the flowfish assay as we measure it and as the group in Vancouver measures it. So you see here, there is this cluster of patients with between 40 and 60, especially males, who all tend to manifest. So I'm going to predict, if I could, that unfortunately the patient who passed away, had we been able to measure his telomere length, he would be in this group below the first percentile. And you see these individuals die out from the population, and you can no longer detect them later on unless they have a lung transplant. This is the major cluster where the lung disease developed. So these people have TERT mutations. The TERT mutations are spread throughout. It just depends on how far the patient's telomere length is from the median for their age. And here, if they are far from the median, they're going to be more likely to develop hematologic phenotypes when they're young. But as they age, the lung sort of accumulates. It's a slower turnover compartment. And as the lung ages, it is more likely to be scarred. And that is the main limitation of lifespan for all these patients, even when they develop cancer. Please let me know or interrupt me, or if there are questions, and we can keep this panel going. But I wanted to show this in this way. Young people who, regardless of symptoms, young people who have these mutations have much shorter telomere lengths. This is the distance from the median. There are basically 4 categories of syndromes that overlap in these patients. And, you know, the patient we talked about, we can classify him here. And if this mutation is deleterious enough, and I think based on the fact that it's in the reverse transcriptase domain and such a dramatic amino acid alteration, I would expect that his kids would be in this category, or potentially even here. So that doesn't mean they will manifest necessarily, but they are more likely to develop disease at a younger age. Dr. Wiesner has a question, I think. Is it in the chat?
No, I didn't put it in the chat.
Okay.
But I'm just really intrigued by, and just to tell the group, Mary helped out with a family that we had, which was similar in a way because the testing itself showed a variant of uncertain significance. And sorting through that, it was very helpful to get the telomere length so that we could really understand what was going on with that particular patient. But could you comment a little bit about how you are evaluating this specific this specific variant with calling it, you know, as having signs of being truly deleterious or pathogenic, and how our laboratories are calling these variants? It just makes it very difficult to test through families.
It's true, and I guess we have the luxury, yes, and we have the luxury of telomere length measurement, Dr. Wiesner, and we have the luxury, I guess, of having stared at this gene. So I can't really make such calls for any other gene, and but this protein is so conserved in yeast that there's very few variants, there are very few polymorphisms in this gene. And we know what they are now after having studied it for 20 years. You know, the family I described was the first reported family with a TERT mutation. So we have catalogs, and when the amino acid is highly conserved and when it's rare, and when especially we have a telomere length result, then I think we feel comfortable counseling for that, however limited the counseling is. So does that help? So I guess, you know, I think that there are in the databases, the classification, you know, as you know, we have as a field tried to organize our efforts, but because I'm a lab-based investigator, and because I've sort of a lot of my career I've spent making sure that this assay is available, we have that luxury. Are there vague cases? Definitely, but a lot of the time we can classify with confidence based on family history, telomere length, and conservation.
So,
Dr. Ross, were you going to say something, or sorry, go ahead.
Yeah, so what I'm hearing is that there's a constellation of other factors that really need to be put into place, and working with experts like you to sort through that needs to be done in this field, really because we're getting the laboratories that are just unable to call it just based on the variant analysis.
based
Definitely.
on the MG criteria, right, Georgia? Like, because that's how they've been classifying it. The other question I had for you, Mary, is when we think of genetic anticipation in other diseases, like the triplet repeat disorders, we think about lengthening, like
Yes.
then, but this is the opposite of that, right? Because it's shortening of telomeres. So it's almost a different, like, do you get, and you explained that earlier, you do get anticipation in these families. And does it matter if it goes through paternal inheritance versus maternal inheritance?
That's a great question. Yes, it does. And we had the reason we have been so interested and keen on these syndromes too is because, my mentor, Carol Greider, who had won the Nobel Prize, or she won the Nobel Prize after some of these discoveries, had generated mice that overlap in many features with these patients. Well before the syndromes were described. And these mice show genetic anticipation because the telomeres shorten over time and they become more and more likely, the mice, to gray earlier and earlier and eventually develop infertility. So did I answer the question?
What maternal versus paternal?
Oh, so the paternal, yes. So in the mice that we studied, we found that later offspring of male mutation carriers had shorter telomeres than earlier offspring. So in our registry, we can see a signal for that as well, where male offspring of TERT or other mutation carriers are more likely to develop disease earlier than earlier offspring. Yes, that is a measurable signal both in an animal colony as well as in our study.
Fascinating. And so would you predict, again, it does sound like this individual, the proband sister, likely has this mutation as well. So would you expect her to have a similar telomere length? Again, not that it's going to be proven, but would that be the expectation?
Yes, I want to take this as an opportunity, as an excuse to sort of talk about the similarities and differences between this group of disorders and Fanconi anemia. So this telomere length test is sort of like chromosome breakage studies in Fanconi anemia. It is our functional test. This group of syndromes is far more common than Fanconi anemia. At least 10 to 15,000 cases in the US alone carry mutations in telomerase, and you will find them most likely in pulmonary clinic, although you all as geneticists are going to come across this at some point. And I want to make sure to talk about MDS before we leave today. And these syndromes, in contrast to Fanconi anemia, tend to have a more, the worse phenotype, an earlier onset of phenotypes in males. So males with lung disease tend to develop disease with the same identical mutation in the same family, tend to develop disease a decade earlier. So I don't know if Dr. Ross's proband was a smoker or not. He wasn't, right?
He did smoke. He quit smoking 15 years before his diagnosis.
Sure, but his sister sounds like she smokes a lot, and she's still hanging in there, even though she has lung disease.
All the sisters continue to smoke, whereas he stopped 15 years ago.
Exactly. So that's even a female smoker might develop disease later than a brother who has the same mutation. So it's something about IPF has a 3 to 4-fold increased risk in males than females, and we don't understand why that is. We counsel on that too, and it informs our screening a little bit, because to your question, and I'll come back to how we manage these patients in clinic, aside from clinical testing, the risk for disease after the age of 40, lung disease, is so high, and a lot of these patients have a very progressive acute course like the one described by Dr. Ross, that oftentimes we want to know about that a priori in order to refer them to lung transplant sooner. So we're very careful to balance the radiation exposure of CT scan once versus the radiation exposure. And we offer, all our asymptomatic carriers, males at age 40, a high resolution CT scan one time. And that, and in females, we offer it a little bit later, 45 years of age, depending on the telomere length. And then we send those people to pulmonary as needed. They don't need to be scanned regularly, but if they have an abnormality at that time, they will likely develop disease in 5 years.
So Mark has his hand up. Mark, you want to ask your question or comment?
Thank you, Tia, and thank you very much, Dr. Armanios. That was excellent. So just going back to the family that Alex presented, I mean, we were being quite conservative because we said this was a variant of unknown significance, suspicious but not conclusive. Hearing you today, you sound more, more in favour of this being actually more likely pathogenic at least. So I'm wondering about the children. I can't remember how old they are, but is there a case for actually testing the children to look to see whether or not they have the variant, and also doing telomere studies at the same time to see whether or not it segregates, with the results that we expect that the telomere segregates with the variant? Because there are 5 of them, so that's a decent number.
Sure. I mean, I think that in our clinic, so we have had a telomere clinic for almost 20 years now, and we've seen so many of these patients. There are different philosophies. As an adult doctor, I feel like if I don't force the genetic testing on children, I think we counsel and we check blood counts when we can. If we're gonna do genetic testing, then we do it with telomere length, not so much necessarily to, prove the pathogenicity of a variant in this case, but to allow us to predict when they will have problems. Because I don't have this slide here, but in this paper, we described how telomere length, the distance from the median, is basically predictive within, with some confidence of when people will develop disease. This is only true for this flowfish-based assay. So then we use the telomere length to decide how to treat, how to screen, when to bring the patients back. If they have a hematologic abnormality on their blood counts, then they're basically stuck with some sort of follow-up. It tends to be a very indolent course usually. And we follow a lot of blood, patients with blood counts, and but we try to stratify based on telomere length and the blood counts in kids, and clinically examine them, And if they look okay, I think we can usually delay the testing until they're adults. If they don't want to be tested, their parents don't want them to be tested. So if I'm giving you a gray answer, I am of the conservative, philosophy that if there's not a clear intervention in kids, we can wait, we can educate, we can follow, we can do some basic clinical screening that is not costly, and wait. Because there's nothing that will prevent the grave consequences except preventing harm and education here.
Thank you. That sounds very reasonable. That's very helpful.
Should I do, and maybe, so this is maybe to clarify a little bit more. This is a paper from my colleague at Hopkins, Kristen Schratz, and we tried to classify here the 4 sort of entities that we see in the primary manifestation. So again, in infants, it's completely different. It's immune deficiency, and these patients have to be transplanted ASAP. This is the 1 in a million presentation. In children, it's this hematologic thing, but as they get older, they are prone to liver disease, a very particular type of liver disease. They may get enteropathies and have gluten sensitivities. they may get avascular necrosis, more rare things, but this is the life-threatening one, along with immune deficiency. And then there's this group in the middle here that has the entire syndrome, and these patients are very difficult to take care of because they may have bone marrow failure and lung disease, or bone marrow failure and liver disease, and they are hard to give multiple organ transplants to at the same time. We have done some transplants in sequence, and our center has a lot of experience in that. And then these adults that need specialized care when they have their bone marrow transplant, if they can get it. And they definitely should not get immune suppression because it can be deadly, or it is deadly. So confusing, and I spend a lot of time trying to convince pulmonologists to have a high threshold for this group of disorders because of the morbidity and mortality of treating these as autoimmune disorders, whether it's the hematologic or the adult onset. So you see why it's not a complicated group of disorders, but same mutation, different generations, different telomere length, causes fairly different manifestations, and but they all relate to one thing, which is the premature aging phenotype. Okay, I think I have the MDS case next. So for the cancer clinics, is that okay to keep going?
Yeah, no, that's great. I just have one other question / comment. So in the NCCN guidelines, TERT is actually mentioned in the AML guidelines. But here's what, like I just pulled it up, what they're talking about, what they mention here is the idiopathic pulmonary fibrosis, hepatic cirrhosis, nail dystrophy, oral leukoplakia, skin hypopigmentation, hyperpigmentation, premature graying, cerebellar hypoplasia, immunodeficiency, developmental delay.
Mm-hmm.
Do those, like, is that comprehensive? Is that correct? Your thoughts?
Yeah, I had input in those guidelines. Maybe they didn't organize them by age, but yes, I had.
Okay, good. No, because they say the blowfish as well. But then in the melanoma guidelines, they talk about TERT, which I don't know where that's coming from.
So I may not get to that because we might run out of time, but I will just tell you that we have been talking about loss of function mutations in TERT for this group of syndromes. But if we don't get to it at the end, there's a very extremely rare group of syndromes that have gain of function mutations that cause long telomere length. And that's where the melanoma comes in. These patients basically never get melanoma. The long telomere patients get melanoma. So it's a sister group of disorders that are basically the mirror image. Okay, MDS. So this is, this isn't a bone marrow failure case. I'll show it real quick. So this was a young man. I think you might see this in your clinics, a young man who was 24 when I saw him. He's had low blood count since he was 10. He has a hypocellular bone marrow, and he was getting ready to be treated with immune suppression. And I saw him and I said, please wait. And he ended up having short telomeres because that would have made his infections worse and his immune deficiency worse. And they were getting ready. Remember, he had no family history. He had a brother who was an HLA identical match, and his brother had normal blood counts, and they were getting ready to transplant him with his brother's bone marrow. And I said, let's check the brother. And his brother also had short telomeres. So that was really, really important. And the primary reason to make the diagnosis in kids in Dr. Ross's family is for a situation like this, where you're trying to identify the appropriate bone marrow transplant donor if they're going to be related. So this is a case never to be missed, because there are cases in the literature from a long time ago where they attempted to give a transplant from a sibling who was asymptomatic prior to when these genes became well known. These are mutations in the telomerase RNA gene, and the patients the graft failed or they couldn't mobilize the patient. And sometimes the graft failed and the patient died. So this is really a standard of care now. And the reason it's important is because telomerase mutations, all of these genes, are probably the most common cause of low blood counts in inherited form. So even though Fanconi anemia has more genes, these genes account for 10% of the inherited forms of bone marrow failure. Okay, yeah, standard of care, you can distinguish the TERT mutation carriers and all the alphabet soup of the related genes from the patients with acquired form of aplastic anemia. And I'll just show you here that the growth, and we're going to exceed these numbers we have in the last 2 years. But most of the volume of flowfish samples in our clinics are patients with bone marrow failure and IPF and prior to bone marrow transplant. I'm going to squeeze this MDS case here. This is a very rare scenario, but a patient I saw also had a woman who had MDS and subsequently developed IPF. And they were trying to find a related bone marrow donor for her, and everyone they checked had low blood counts. And this patient ended up, my colleagues at Hopkins didn't recognize this was a telomere case. And they referred to the University of Chicago. It came back as a TERT mutation. And this is where I wanted to leave you here, that only 15% of patients with telomerase mutations develop an AML MDS. This is a spectrum. This patient eventually died from acute leukemia. So this is a spectrum, and these disorders go together when they run in families. But this is TERT mutations, I think, in my interpretation of the literature, are the most common cause of inherited forms of MDS. But look, it's much smaller than the IPF group. And it's rare enough that you can't screen for it. And this is what Dr. Paul just mentioned in the NCCN guidelines. So if you have a patient with AML and a family history of IPF, that patient should be tested, should be evaluated. And we can talk about the source of germline DNA. And this is a TERT, among patients with familial forms, more than one family member with MDS or AML alone, this is the most common cause. Telomerase mutations in general in all their forms. Okay, we have 2 minutes. Are there questions maybe we can take rather than rush, or what should we do, Dr. Powell?
So what I'm wondering, this reference looks like it has a lot of the stuff that you have gone through. So maybe what we'll do is circulate the reference to the group. And are there patients that we should be considering sending to you? Again, it sounds like Dr. Ross's patient the sister might be a good one to send telomere.
Sure. I think I'll just say, in telomere length testing for those of you in the US should be very accessible. It's a relatively cheap test. It's $500, $450, and the turnaround is 2 weeks. So I think the main indications are the ones we went over now. And if you are in a place overseas that doesn't have access and you have a patient, please, find me and we'll help you get the patient tested, or we can help you interpret accordingly.
Can I ask another question? Why is it so few labs do this testing? Like, why are you the only one in the US that
it's a very laborious test, and it requires a lot of care, and it's not NGS based. So many companies had tried to develop this assay and sort of gave up, and it was through a very perseverant postdoc in my lab who was interested in flow cytometry. And as we came to realize the value of this testing, we sort of just invested time into making sure that people have access to it in this country and hopefully beyond. It was it was an attempt to take advantage of the science in this case to help patients.
That's amazing. Georgia, did you have another question?
I have many. But so I'm kind of pondering. I really like the slide that you had of the ages and what you might see. And so I think that would be really helpful for us to think a little bit about as patients come into us, what we might do. The other thing I'm really learning again is we need to go beyond our clinical genetics paradigm of DNA testing through the family and making sure that we think of other tests that might be able to help us make a diagnosis. And that is different. That's hard for geneticists to, rethink about that. So that would be just one of my take-home messages that I'm getting. And Tula, you're saying that you're going to be able to share some of the papers so that we can follow along. It would be very helpful.
Yeah, no, that's what I'm thinking. Mary gave us that reference, so we can pull that paper and share it.
Really helpful.
Sure, I'll send the one with the discrete manifestations also.
Mm-hmm. Yeah, that is just really helps frame it as you come in. And parenthetically, I had a patient yesterday who has myelofibrosis. And a number of things found on NGS. And so now you're making me go back and go, okay, well, do I need to do more for him? So thank you. Very timely talk.
Well, myelofibrosis is a bit different. For some reason in short telomere patients, the fat takes over the bone marrow, not the fibrosis. So it's different, but that's another group of disorders which we need to understand more for sure.
Yeah.
Thank you for what you do in bringing everybody together and in having a forum to discuss cases like this.
Yeah, no, thank you so much, Mary. This was really, really fantastic.